Okay, this meeting is recorded in YouTube.
Good afternoon, my dear students. I welcome you to this online live Zoom class for unit test 9. We have two chapters. One is evolution, another one human health and disease. So now we are going to discuss first the evolution, and I will move on to the next topic. So you know that evolution is a process of the existence of the highly complex organism from the simple primitive organism, that is called evolution. Here first the origin of life. We know that the universe is very old. The universe existed about 20 million years ago. Okay, 20 million years ago, so it was very old. And this universe consists of many galaxies. They consist of many galaxies, right? So in the primitive, there is Earth that is developed about 4.6 million years ago. The Earth originated about 4.6 million years ago. And here, this theory called Big Bang theory that helps to know more about the origin of universe. The name of the theory called Big Bang theory. It's called Big Bang theory. Okay, that helps to know about the origin of this universe. So in the primitive, actually this universe expanded. So due to expansion of this universe, the temperature got reduced or decreased. Okay, this is slowly decreased. But probably the primitive Earth, only the two gases are present. So one is hydrogen. Another one is hydrogen. Another one is helium. Only the two gases are present in the primitive Earth, and slowly the temperature also got reduced. But there was no atmosphere in the primitive Earth. Right? In the early Earth, there was no proper atmosphere. There was no oxygen, no carbon dioxide. Only these two gases were present in the primitive Earth. And life appeared. Life originated approximately about 500 million years ago. Okay, 500 million years ago. Sorry, the Earth originated about 4.6 billion years ago. 4.6 billion years ago, but the life originated about 500 million years ago. So nearly 4 billion years there was no life on Earth. The duration we call this a Mesozoic era. It's called a Mesozoic era. So there was no life from the birth or formation of the Earth till the first life originated. And the various the scientists they given a different ideas and concept about the origin of life. Like special creation theory that is proposed by Father Suras. According to him, the Earth or the whole universe and also all life were created, were developed only by God. Like special theory, this is called a special creation theory. And Cosmozoic theory, Big Bang theory, abiogenesis, and biogenesis. So the different scientists proposed the different theory regarding the origin of life. But for all this, there is no direct evidence to prove, right, that the life originated from their, I mean, the sources what they have mentioned. According to, the Hindu belief also, the God Brahma created everything. And according to this Christianity, Jesus, God created everything. So like, God created. So like, these concepts actually, there is many direct evidence to prove this. So the final and also all accepted theory proposed by Haldane and O'Brien. Okay, Haldane and O'Brien in the year 1928. The theory we call is the chemical evolution. The theory proposed by them is called a chemical evolution. Okay, chemical evolution. And according to them, here in the actually they proposed 1928. According to them, the primitive Earth only the four gases. That is methane. Okay, CH4 and ammonia. H2 is the water vapor, not free oxygen, and also hydrogen. Only these components were present in the primitive Earth, according to Haldane and O'Brien. So, according to them, because of the high temperature, this is also another important one. The high temperature, okay, and volcanic eruption, and ultraviolet rays. Okay, because of these, the environmental factors, methane, ammonia, water or water vapor, hydrogen, they had a reaction and they produced the first organic component. So Haldane and O'Brien, right? They believed that the life originated from these chemical substances. So they call this, they proposed this theory called the chemical evolution. So this theory were proved by the experiment done by or supported by Urey and Miller. So look at this picture. Is a Urey-Miller apparatus. So this, they tried to produce, they tried to develop the organic component. So in their apparatus, they took the methane, ammonia, water vapor or water, hydrogen in a, this chamber. This chamber is a large, is a spark chamber, and in this spark chamber, the two electrodes were fixed. The electrodes that helps to produce or develop the lightning. So the experiment is also called stimulation experiment. The experiment is also called stimulation experiment. The stimulation experiment, they're done by Harold Urey and Miller, right? And here, in this, the temperature were maintained about 800 degree centigrade. So at the one end, this one is connected with another small chamber, and in small chamber here it is constantly, it was heated. Here the water were taken, and because of heating the water vapor, right, there is a supply to this spark or this large chamber, right? And the opposite end, it connected with the U-shaped, trap. Connected with the U-shaped trap. Before it connected with this U-shaped trap, here the condensers are also fixed. Here is the condenser, also fixed. In this condenser, the water were allowed in, and also the exit out, mainly to reduce the temperature. So inside this chamber, the temperature was around 800 degrees centigrade. That is, it is created like as like this, the primitive Earth. The temperature were given or increased or is maintained up to 800 degrees centigrade. When methane is ammonia, water vapor, and also hydrogen, they had a reaction with one another. in this high temperature, and when it is constantly, it is allowed to circulate, right, or allowed to pass into this U-shaped cracker. Before that, the condenser in this, they passed through this condenser, so that the temperature got reduced, right, in this area. So that the content of the substances were evolved during this reaction. All this were reaching this U-shaped tube, and here it is collected in this the liquid water. This trap collected, and the substances were analyzed. The substances were analyzed, and the first amino acid. So Urey and Miller, they obtained the amino acid. So the first amino acid they obtained was glycine. Right, first amino acid got glycine, and then we got alanine and also aspartic acid. Okay, aspartic acid. So they, okay, they got this glycine, alanine, aspartic acid, 3 amino acids, and actually they took this methane, ammonia, and also this hydrogen. Methane, ammonia, hydrogen in the ratio of 2:1:2. This also you have to concentrate in examinations. Listen, here you must remember the temperature 800 degrees centigrade is the important one, and also the name of the amino acid, right, or the amino acid they got in the experiment is another important one, and the ratio of this methane, ammonia, and hydrogen 2:1:1. This is also important in Neet. You have to remember these points. And then the similar experiment done by some other some scientists. Their similar experiment, they got the different components, especially they obtained sugar. They got sugar, okay, sugar, and the nitrogenous base, and also some pigments, fat, okay, fat. So the different components were obtained. By some other scientists, they did the same kind of experiment. So from this experiment, it's clearly we know that the life originated from the chemical substances, not from the pre-existing life, or not created by God, right? Not created by God. So this process we call this a chemical evolution. And after the simple form, there is a development. These amino acids as well as the sugar, nitrogenous base, pigments in the fat is were developed. These are actually non-living, and these substances they have a reaction to one another, and they produced, they formed this protobionts. They formed all these other reactions, right? The constant supply of energy they obtained using the solar energy. So there was no ultraviolet radiation, no ozone layer. So ultraviolet radiation reached the Earth, and they had a reaction in the high temperature, and they produced the protobionts. Okay, protobionts. These protobionts, right? The characteristics. This is another important one. They do not have the ability to reproduce. Okay, the lack. Okay, reproduce. The lack reproduce. So they do not have the capacity to reproduce, and the other components. So the sugar and the nitrogenous base, and also the amino acids, they had a reaction to one another and produced quiescent. The quiescent also do not have the capacity to grow. Okay, sorry, they can grow, they exhibit metabolism, but these do not have the capacity to reproduce. So protobionts is including quiescent, microsphere, okay, and vesicles. So these three, we call this the protobionts. They do not have the capacity to reproduce. But they exhibit the metabolic process, and they have the characters to grow. They have the characteristics to grow. And later, from these the primitive substances, the complex organisms are complex disease. The next level of organisms is the development. Right? So these three do not have capacity, and after the RNA that discovered by Hercule Purana, and the first nucleic acid structures like RNA were evolved. This RNA have two characteristics. One, the RNA able to act like as an enzyme, right? It is called a ribosome. It's called a ribosome, and there is a catalyst, and also the RNA it act like as a there is a genetic material also. Okay, act like as a genetic material also. So life existed from the non-living things only, and later the anaerobic. Okay, there is a anaerobic bacteria, heterotrophic bacteria were developed. From the heterotrophic bacteria, chemosynthetic and photosynthetic autotrophic organisms were developed. After that, the oxygen, the carbon dioxide evolved in the earth. So in this chapter or in this part, we must know about the Urey-Miller experiment. That is an important one, and the diagram, and also you must know the components, the ratio in which they have taken. All these things you have to remember. Okay, next, what are the evidences supporting this? The evolution, and here the evidence is, look at this. The evidence is for evolutionary process. Here the organs, organs we call this the homologous organs, analogous structures, and vestigial organs, the various connecting links. All these are supporting the evolutionary the events or evolutionary process. Here, homologous the concept of homologous terminology that is proposed by Richard. Okay, Richard Owen. His name is called Richard Owen, and according to him, right? According to him, these homologous organs. If you look at this, these these are bat mammal, and here whale is an aquatic organism. These are past running animal. Is a human being, right? In these all organisms, you can see the basic pattern of the bone structure in the forearm region. This is a forearm bone. Here in human being, the long bone we call this forearm bone is the humerus. It's a humerus bone. Okay, humerus bone, right? And the radius as well as the ulna, and also here the is a carpus, metacarpus, and the phalanges bone. The basic pattern of these four is similar in all organisms. Because they are living in different habitat. It's a fast-running animal, but the whales are living in water. This aquatic organism. So in this, the organisms developing or possessing, the basic pattern of the structures for origin and development. Origin and development of organ, that is the similar pattern. Okay, similar pattern and the similar structure, but they have different functions. So if the organisms are developing, or the organ is same and the function is different, this kind of organs we called homologous. It's called homologous organ. So here we must know many examples. That is important. We must know many examples of homologous organs. So these homologous organs, when diversified in the sense with the same pattern, same origin, when they are living in different habitat, this leads to the divergent evolution. leads to divergent evolution. Okay, this is so homologous organ leads to divergent evolution, and you must know the examples. Sometimes they give only the picture and they will ask, the four options like this. And imagine, remember this: the homologous organs, development of the homologous in the organism that leads to the divergent evolution because they adapt to live in different habitats. Here, the two examples that is given. One is in the animals, another one in the plants. So if you look at this, the bougainvillea and also the cucurbita, here is the node region. In node region, actually the structure called thorn that appears, but here it is the cucurbita, the tendrils, right? The tendrils, okay, that helps to support. It helps the plant because it's a weak stem plant. This tendril helps the plant to support with any object so that the plant can grow well. But here in bougainvillea, this instead of tendril, the thorn is present that gives protection, right, from the grazing animals. So tendril for climbing and the thorn for the protection. Okay, thorn for protection. So like the origin is actually same, but they have different function that you have to remember. So in addition to that, there are many examples are there. For example, there is the mouth parts of insect. Okay, mouth parts of insect. So origin is same, but in some cases the mouth parts that is useful for getting food, especially in butterfly, proboscis-like structure is there for sucking and siphoning. But in the case of housefly, it is a sponging type of mouth parts. In the case of mosquito, it is a piercing type. So the mouth parts are similar, but they have different function. And like potato and also ginger. Potato and ginger. Another example is also we can say potato and ginger. These two are the modified stem. This is a modified stem and limbs of cat. Okay, limbs of cat, lizard. For walking. These limbs of cat and lizard for walking, and the limbs of the aquatic organisms like whales. Limbs of aquatic organisms like there is a whale, right? Whale and the bat. So these are some of the examples of the homologous organs, right? And another one is the analogous structure. Analogous structure, you remember this. The analogous organ is here the example is given. The wings of insect. The wings actually appears from the developed from the thoracic segments. The pterodactyl is the Pterotaurus, a dinosaur. Okay, a dinosaur has become extinct. This one is the bird's wing and the bat's wing. So the basic pattern is actually different. The basic pattern is different in the case of analogous organs. This example for analogous. Okay, analogous organs. The basic pattern is different, origin is different, development is different, but the function is same. So the function is nothing but useful for flight. So they are living in different habitats, including the extinct organisms, right? Like a pterodactyl, you can see. The origin is different, but the functional, function is same. So this kind of the relationship or this kind of this organs we call is analogous organs. So these analogous organs is found in this case of insect, that is pterodactyl, bird, and also bat. In addition to that, you must know some more examples. So different structures evolving for same function is called analogous organs. Different is for structure and but a similar function is called analogous. So the other examples like wings of butterfly. So in examination, the other examples also can ask. So you have to prepare for this also. Wings of butterfly and also birds and birds and some other examples like flippers. Okay, there is a flippers of penguin and also dolphin and sweet potato and also potato in plants. So these are some of the examples of the analogous structure. We have to remember these organs also because this important in this examination point of it. So the analogous organs, it actually leads to convergent evolution. Convergent evolution. Right? Convergent evolution. So convergent evolution, the origin is different, but the function is same. Okay, function is same. And another one, we come to this vestigial organ. Vestigial organ. So the vestigial organ, okay, vestigial organ. Here, these organs were functional in our ancestors. Functional were in our ancestor, but is non-functional or become a vestigial structure without any role. Not performing any function. These organs are called vestigial organs. So they are not performing any functions. Actually, there are about 86 vestigial organs that is existing or present in human being. Right? 86 vestigial organs are present. So this is also one of the evidence that the organisms like human being evolved from our ancestral, the former ancestral organisms. So, for example, the nictitating membrane. So, some of the examples: nictitating membrane, or it is also called the third eyelid, and ear muscle, ear muscle, and the wisdom teeth, okay, and the muscles present in the muscles, okay, in forehead region, and coccyx bone. Okay, this is a coccyx bone, and appendix, vermiform appendix. Vermiform. Like these are some of the examples of the vestigial organ which are functional in our ancestor. Better, it is it is present. The organ is present without any role or without any function. And the hormone called MSH, melanocyte stimulating hormone. These are the some of the examples of the vestigial organ present in human being. So these also indicates that, right, that a human being evolved from the pre-existing organisms, right, or the the underdeveloped or primitive organisms. And another example that here embryological evidences. If you look at the diagram, you see the organism of the embryo stage. Here it is an embryo of, this is an embryo of this embryo of fish. This is an embryo of fish. This is an amphibian. Okay, this one is the chick or is the bird, and the last one this is the embryo of this human. So you see this one fish. So picture is given. So look at this. So this is the embryo of fish. embryo of salamander, is a tortoise, a reptile, is a bird, and also human being. If you look at this, in fish, the gill slit is present. The same kind of gill slits are present in all these organisms, in all these classes, because human being is an advanced the group of organism, but during embryonic stage, the gill slits are present. And slowly, when it becomes the mid-embryo stage, these gill slits, that is disappears, but the limbs appears. The appearance of limbs appears only in these tortoise and birds and also the human being, right? Like from primitive form, or if you look at this in the primitive form or the embryo stage, right? Some organs are evolving or developing, but during the latter stage of embryo or in the mature embryo, these primitive structures like gill slits and also the appearance of tail completely disappears. Instead of that, the highly developed limbs are developing, right? Both fore and hind limbs are developing in the embryo. This indicates that. So during embryonic stage, all the species of organisms shows their ancestral character. It shows their ancestral character. So this theory is called, actually this is proposed by Ernest Haeckel. Theory proposed by Ernest Haeckel. Okay, Ernest Haeckel. The theory is called biogenetic law. Biogenetic law. According to this theory, according to biogenetic law, according to this theory, every organism shows their ancestral character. According to this, every organism showing their ancestral character. So this is also another important one to meet. This theory proposed by this man. That is the theory. Original name is called original name of theory. It is called ontogeny recapitulates phylogeny. This is ontogeny recapitulates phylogeny. That means during developmental stage, they shows their history of their ancestors. They shows their history of their ancestors called ontogeny recapitulates phylogeny. So this is another important. In addition to that, the connecting link also you have to consider. Connecting link. Okay, connecting links. The connecting link, actually the organism possesses the characteristics of two groups of organisms. Two groups. For example, peripatus. They shows the characteristics of Annelida and the arthropoda. So the example you have to remember. Okay, and neoplenia. Neoplenia is shows the characteristics of Annelida and mollusca. Annelida and mollusca. And chimera. This shows the characteristics of both the conflict species. Convictis and ostrichis. Okay, convictis and ostrichis fishes. And the familiar example that you know, Archaeopteryx. Okay, Archaeopteryx, a connecting between birds and reptiles. Connecting between birds and reptiles. And the example, the connecting between reptiles and mammals. Reptiles and mammals. Okay, that is prototherians. Prototherians. Like these are all examples, right? You must learn more examples than this. So, like these are some of the evidences that organisms that is developing, right, from the pre-existing one. So that's why some group of organisms possess the characteristics of the two different phylum or two different group of organisms. Is also you must remember and go through some more examples. And the atavism characteristics. In atavism, sudden appearance of the parental or ancestral character, like appearance of tail. Okay, appearance of tail in babies is another example. Okay, appearance of tail in babies is another example for atavism character. So all these evidences that indicates that the present day organisms are developing or evolving from the pre-existing one, but the life developed or originated by the help of these chemical substances. Right? So next one, the adaptive radiation examples. Adaptive radiation example or evidences. Okay, so this one in the picture, you look at this, is a Darwin's pictures. You know that Charles Darwin, when visited the Galapagos Island, he found the morphological differences in the finch, and actually the ancestral finch is where probably they had only the fruit eaters. Okay, these the primitive organisms were fruit eaters or seed. Okay, these were the seed eating. Okay, they had the seed eating characteristics. So Darwin found there are about 14 different. Okay, how many? 14 different species of finches. 14 different species of finches in Galapagos Island. Right? 14 different species. Among these, the last one you look at is warbler finch. Okay, that is the smallest finch. Warbler finch. The examples also, these are actually all these are vegetarian warbler finches. Okay, sorry, vegetarians. These warbler finches are smaller in size, and the vegetarian tree finch, this one is the largest one. So if you look at this, you can see according to its the food habits. So some they feed on insects. Some finches they got food from the cactus plant. Some were the seed eating. They had a seeding habitats, and here insect-borne these finches also had new modifications here, and these are feed on the plants or the plant parts. Later, depending upon its food habits, the beak modification took place. So how the beak modification? According to the necessity in the environment. The type of gene which is present in these organisms, it is called ALX1 gene. The name of the gene called ALX1 gene. That leads to the phenotypic changes in the shape of the beak. Actually, this gene produces a type of protein. It is a protein that is called beak morphogenetic protein. Beak morphogenetic protein. BM. Okay. BMP. Right? These genes are changed according to the need in the environment. Right? They cause change in the craniofacial structure. I think this change in the craniofacial structure. So that the head as well as the appearance of beak is totally modified. This is a good example for the adaptive radiation. The another example, if you look at this, there are two more examples. This is a name you must know. So the ancestral marsupial animal were radiated and developed into the different it is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 different type of the organisms. So this is another good example for it is adaptive radiation. So they developed new characteristics and they able to live successfully in the new environment. So from the ancestral form, the 10 different types of these organisms were evolved, 10 different species were evolved. This is another good example for this adaptive radiation. You must know all these names. Okay. There is an example on the match the following paper. They are expected in asking this kind of question. So good example for this adaptive radiation. Another example here it is given now the difference between these placental mammal and also Australian marsupials. Placental. So the placental mammal, the one which is living in burrow is the mole. But here marsupial mole is also there. But here this is actually example. Actually again more than two different. The divergent evolution. More than two different. For example, from here one type of organism is from from ancestor form, one organism is deviating and living on habitat. And here from habitat it is they are selecting another habitat. So and third species like this. When these are preparing the same particular habitat, here the characteristics become similar. So here one example for adaptive radiation or divergent evolution. There is second example for divergent evolution. Third example for divergent evolution. If more than two divergent evolution that prefer the same ecosystem or same characteristics, and they are getting the same kind of habitat as well as characteristics, this kind of adaptation is called adaptive convergence. It is called adaptive convergence. It's called adaptive convergence. So this is also an example for adaptive convergence. For example, placental mammal is different from the marsupial. The marsupial mole. Okay, but both are preferring the burrow to live. Right here, number is anteater is a different one, different species. But here the anteater, these two are having the same characteristics. So like more than one species, right? More than one species that prefer one species in the sense they are originated from the common ancestor and preferring the same ecosystem, either the place or mode of nutrients, whatever it is. And when more than two or three adaptive radiation that actually prefer the same ecosystem or same kind of food habits that leads to the adaptive convergence. It's not adaptive radiation. It's called adaptive convergence. This also leads to, remember this, in examination, the adaptive convergence leads to the analogous organ. Okay, this leads to the formation of the analogous organs. This is another important one. Remember this. Analogous organs develop in the organisms because of the adaptive convergence. Adaptive convergence due to the more than 3 or 4 or 2 or 3, 4 group of organisms they are diversified and they possess the same characteristics. This leads to adaptive convergence and also formation of the analogous organs is also you have to concentrate. Right? The example and also name that you must know. Okay, so next one, some of the examples that you have to remember. I will come to this later. This is a diagram. Before we go into that, here the Hardy-Weinberg law, another important one. Hardy-Weinberg law. Okay, Hardy-Weinberg law. So according to these are two scientists, the relative frequency of an allele. You know that allele is an alternative form of the gene. Alternative form of the gene. In a homologous chromosome, either a 2 dominant gene or 2 recessive gene or 1 dominant, 1 recessive may be present. In a homologous chromosome, if you take a human being, we have 46 chromosomes. To determine a particular character, both the chromosomes may have dominant gene and both may have the recessive genes or 1 dominant, 1 recessive may be present. So the relative frequency of this gene, right? This gene. Actually, the dominant gene is expressed as a P and the recessive gene is expressed as Q. Right? The A gene is expressed right as a P and the recessive gene Q. So this frequency of this gene is always constant. Always constant in a large randomly mating population. Large randomly mating population, this dominant and the recessive allelic frequency is constant. Suppose if the various factors affecting this equilibrium, here this leads to the variation and new species of organisms will be developed. So in the absence of mutation, genetic recombination. So the 5 points that you have to remember, one is gene migration or gene flow, gene migration, right, or gene flow, and gene mutation, and chromosomal, genetic recombination. Okay, gene migration or gene flow, genetic mutation, genetic recombination, genetic drift, and natural selection. Okay, natural selection. If these factors are not affecting, the allelic frequency in the population is always constant, right? So here, I will give you some examples and I will give you some question also. So normally, the allelic frequency, right? Allelic frequency, that is P plus Q equal to 1. But the frequency, okay, genotypic frequency is P square plus 2PQ plus Q square. Right? P square plus 2PQ plus Q square. Right? And here, this actually you must know this the two formula. For example, the allelic frequency of dominant gene and allelic frequency of recessive gene, right? That is giving this value like 1 in a large randomly mating population. That is a allelic frequency, or is the expression of both dominant as well as the recessive. Okay, that is alleles. But in genotypic frequency is a P square plus 2PQ plus Q square. I'm giving a simple example. Frequency of dominant allele. The frequency of dominant allele is about 60%. Okay, and sometimes they ask what is the percentage of, find out the percentage of heterozygotes. Right? So the P is actually 60. So it is 60 by 100. So the P value is 0.6. And from this, so how can we find out the P? Q equal to P minus 0.6. Sorry, 1 minus. Okay, 1 minus 0.6. So the Q value is about 0.4. So from this P value and also this Q value, we can easily find out this PQ value. So the PQ value is the genotypic frequency is about 2 into P into Q. 2 into P into Q. So 2 into P value is 0.6 and the Q value is 0.4. So here, so 0.24. So 0.24 into 2, it is a 0.48. So the genotypic frequency is 0.48. So if you total up this, the dominant gene frequency and the recessive gene frequency, right, that will come one. That is also, remember this. So few more examples that I will give you in assignment questions. Right? You go through this. Also, there is another important one. This what is it? Hardy-Weinberg law. Okay, Hardy-Weinberg. Okay, next one. Next one. This look at this. This example for different types of selection. Different types of selection. So selection is a okay, this selection here. This is the original population. You look at this graph. This is the original population of, right? This is a fur color of the. So examples I am giving you the rat, fur color of the rat. In original population, here from the white albinos to the dark colored organism that is successfully living. But how the natural selection operates in the population? First, you see this. First one, you see the stabilizing selection. In stabilizing selection, the combination with the environment is not very severe. The combination is not severe. The selection operates in the stable environment. The selection operates in the stable environment. So here, the characteristics, that is a phenotype 2, it is a what is it? The dark colored organism that is able to live in a natural or the original population. But in the selection or the stabilizing selection operates. Stabilizing selection operates here. The organisms which are possessing the the deleterious phenotypic characteristics present at both the end of that is completely removed only the characters which are stable, are the average phenotypic characteristics. What is it? The average phenotypic characteristics alone is selected. The deleterious characteristics at both the end. So that means the albinos are deleted, and also the dark circular organism also that is removed from the environment according to the stabilizing selection. But here the combination is not very severe. And then come to that another one, directional selection. In case of directional selection, from one end that is to possible extremities is the original population. Up to this possible extremities, the phenotypic characteristics are altered, but at the maximum, when it is reaching the maximum, the previously existing phenotypic characteristics disappears, and the population evolving a new character. New character in the sense that the dark colored characters evolved, or the organism able to change the characteristic up to the possible extremities to survive, and the remaining characteristics organism disappeared. That is the case of directional selection. The third one is a diversity selection, or it is also called a disruptive selection. It's called a disruptive selection. The disruptive selection are always encouraging the organisms which are in only the extremities, but the moderate characteristics organism disappears. So imagine if this particular character possessing organism, phenotypic organism disappears, they are evolving as a two different species. So diversifying selection or disruptive selection encouraging the polymorphism, or or it is also responsible for the origin of more species. Then now here the population is split up into two different species. So this is another the important one. The diagram is important for this the types of selection, and we concentrate more on this also. And when you come to this what is it, melanism or industrial, because of this industrial evolution, is also experimental verification of natural selection, and is two different species. One is called the insects or the moth, right? Or it is called the peppered moth. Peppered moth, which was gray in color, gray color peppered moth, is called the distant butyl. Distant butyl. And the black one is called distant carbonyl. Distant carbonyl. So these two populations were predominant in England before industrialization, but especially in Birmingham city after industrialization. So these grey-colored moths were easily caught by their enemies, and its population disappeared. Only the environment selected the Butyllaria carbonaria because the color of the insect or color of the moth is more or less color of the stem, color of the stem. So because of the pollutants released from the factories and industries, they completely, that is the soot, okay, released from the industries, they completely change the color of the stem. So the organisms which have modified themselves to live in the particular environment is able to live successfully in the environment, but the remaining organisms remain in the sense the white color or grey colored moth were easily caught by the enemies. So the experiment done by the scientist called Bernard Kettlewell. His name is called Bernard Kettlewell, and he actually introduced reared both the species, and these allowed to grow in the polluted area in the Birmingham city, right? Birmingham city, and one more group that is released in the unpolluted area, that is Dorset city, and after completion of its life cycle, he collected the insect, and he found the Carbonaria species were predominant in the polluted area, and the Dorset were predominant in the unpolluted area. Actually, he released both the types in both the environment. So nature selected in unpolluted area the grey moth were predominant, in polluted area dark this Butyllaria carbonaria were dominant. So from this experiment, he proved that the organism changing their characteristics according to the environmental conditions. So here also we have to remember these the examples and also how they are showing, right, the modifications changes according to the environment. And another one, that is, look at this picture. There is the evolution of man. Evolution of man. Here the human being actually evolved. The present human being called Homo sapiens evolved from the monkeys. Evolved from monkeys. So about 15 million years ago. 15 million years ago. The Dryopithecus. Okay, Dryopithecus. They lived the Dryopithecus where there is a common ancestor of both apes and also man. They lived 15 million years ago. Common ancestor of both ape and man, and they had a hairy body. Okay, and they walked with 4 legs. Okay, 4 legs, quadruped locomotion. It's called a quadruped locomotion. And from this Dryopithecus, the next the Ramapithecus evolved, and Ramapithecus also evolved about 50 million years ago. 50 million years ago, and a hairy body again. Okay, hairy body, right? And 4 feet height. 4 feet height. That is the characteristics of Ramapithecus, and found in the fossils are found in the Shivalik hills in India. So next, Australopithecus, actually these fossils are found. They lived 2 million years ago. right? East Africa, and the first bipedal. First bipedal. Okay, that is they walk with the two legs. First bipedal and the first ape man. Okay, called first ape man. Right? And Australopithecus carrying capacity is about 300 to 500 cc. So up to Homo and Neanderthal men, their carrying capacity was very less than 1000 cc. And next is the Homo habilis. Homo habilis. The habilis meaning is mentally able. Okay, mentally able. And the first human like this man is a tool maker. Okay, and handyman. They are also called the handyman. So here you must know this cranial capacity. The cranial capacity of this man is about 650 to 800 cc. 650 to 800 cc. And Homo erectus, that is about the cranial capacity is about 900 cc. They lived about 1.5 million years ago, and they ate meat. They were the first meat eaters. Homo erectus were the meat eaters, and they used fire. They used fire. Okay, first they learn how to use the fire. They are also called the pecking man or Java man. This Homo erectus is called a pecking. Okay, pecking man or Java man. Pecking man or Java man. This is also an important one. And Neanderthal men, they lived in Central Asia. Cranial capacity is 1400 cc. 1400 cc. So they lived about 40,000 years ago. 40,000 years ago. Okay, 40,000 years ago, and they learned how to bury their dead. They actually had a religious belief also. Religious belief. They used the skin to hide themselves. Right? They wear the dress by using the skin of animals. And the one more man is not given, that is Cro-Magnon. Cro-Magnon. They had brain capacity. It is 1,600. The 1,660. 1,660 cc. Okay, the brain capacity they had. Right, and they are called a Cro-Magnon. They lived in they lived about 30,000. Cro-Magnon lived 30,000 years before. 30,000 years before. And Homo sapiens, they originate about it is 10,000, 75,000 to 10,000 years ago. So they are omnivores, and also they developed during this. They formed evolved during the Ice Age. They evolved during Ice Age. So here, the cranial capacity is an important one for one more question. That is a neat question. So the evolution of human being, that is we are calculating based on their cranial capacity. Based on their cranial capacity, and here you must remember human being, of course, omnivore organisms, and 18,000 years ago they lived in caves. Okay, they lived in caves. So up to the human evolution, we have seen a little bit. So, dear students, I will discuss some of the questions. From this lesson. In addition to that, you have to prepare this. Okay, human diseases, including smoking, and the structure of antibodies, cancer biology, viral as well as the bacterial fungal diseases. So you concentrate more on this. I'll give you 10 minutes break. After the break, we'll discuss some questions, and after that, I'll give you preparation and I will send you the assignment question also. Thank you. Stay here and we'll continue after 10 minutes. Thank you. Okay, listen, students, just to look at the questions, we'll discuss questions for evolution. The remaining, go through that. See the questions. First one, what was the most significant? Okay, it is a trend in evolution of man. There is a significant trend in the evolution of man. Not in the upright posture, not the shortening of jaws, not binocular vision, because binocular vision is there in lower organism also. Increasing brain capacity. Okay, in Homo sapiens, the increasing brain capacity, right? That is a significant in the evolution of modern man. Next question. The particular locus, the frequency of A allele is 0.6 and that of 0.4. Okay, small a is 0.4. What would be the frequency of heterozygous? Normally, randomly mating population. See, A allele is equal to 0.6. That is P equal to 0.6. Small a is 0.4. 0.4. So here the formula is P square plus 2PQ plus Q square equal to 1. Equal to 1. So the P square is about the directive is given. It's directly given. P value is 0.6. Q equal to 0.4 is directly given. So P into Q, right? The formula is 2PQ equal to 0.2 into 0.4 into 0.6. So it is about 0.24 into 2, but 0.48 is the answer for this question. Okay, so 0.48 is the correct answer. And the third one, evolutionary convergence is the development of evolutionary convergence development of is a common set of characters. Common set of characters in a group of different ancestry. Different ancestor, but they possess the common characteristics called evolutionary significance. So option A is the correct. The next one, in the case of evolution of tapered moth, distal butyl area, black color from became dominant over white color from England during industrial evolution. That is because of the not appearance of dark color individuals due to very poor sunlight is not a protective mimicry. It is also not inherited character, and it is a natural selection. Okay, because the environment is dark, so the nature selected only the one which possesses the dark colored wings. Fifth one is also another environment is a horse evolved. There is a progressive reduction number of toes. Right? So which toe is become as a hoof? Third toe. Third toe is developed as a hoof in this case. So here evolutionary order. So Hyracotherium, remember this Hyracotherium here in 2 is the ancestral form of the present horse. Hyracotherium, or it is also called Eohippus. Okay, it is also called Eohippus. Hyracotherium or Eohippus. So from the Hyracotherium, Orohippus, Mesohippus, Merychippus, Pleohippus, and Equus. This one is correct answer. So you go through this evolution of horses also. So the modern horse is called Equus. Ancestral form is called Hyracotherium. The earliest, next one, earliest was human ancestor from India. From India, Ramapithecus, from Shivalik hills. From Shivalik hills, that is Ramapithecus. To be evolution of successful, a mutation must occur. When mutation occurs only in germ classes, then only germ classes is nothing but the gametes. Now occurs here in this that will be inherited. Suppose it occurs the somatoplasm, only the changes will happen in the particular organism only, not inherit to the next generations. Study of fossil. Study of fossil is called paleontology. Study of fossil is called paleontology. So, only the fossil evidences are supporting, right, these evolutionary processes. And the next one, which are the following fossil man proceeded training capacity equal to the modern man? Equal to the modern man. Modern man is 1000, that is 1400 cc. 1400 cc in the case of Neanderthal. Neanderthal man. In others, in all these three, the training capacity is less than 1000 cc. Next one, which are the following is related with the reproductive isolation? Reproductive isolation, there is gametic isolation. There is a gametic isolation, temporal isolation, behavioral isolation. All three are related to reproductive isolation. Question number 11. The concept of population will increase geometrical ratio. Of course, the population is actually increasing more in geometrical ratio than the food production that was given by this Thomas Malthus. This theory is called demography. It's a demographic, not a Hill, not Smith, Charles Darwin. Next one. What kind of evidence is suggested that a man more closely related to chimpanzee? These evidence is from DNA, not from the sex chromosome, both the chromosome. Allosomal chromosome as well as autosomal chromosome and mitochondria. It is similar in chimpanzee as well as in man. Next one, the chemical substances formed in Miller's experiment was not nucleic acid, amino acid. Okay, amino acid that is developed. Now the three different types of amino acid developed. Remember, first is the glycine, alanine, and aspartic acid. Glycine, alanine, aspartic acid. Lamarck's theory of evolution. Okay, his theory is called not special creation. Special creation theory proposed by Father Flores. Survival of different tests given by Darwin, and inheritance of aquatic character. The example, midcline of giraffes is the example for this Lamarckian concept. And which is not true of Archaeopteryx. Archaeopteryx is a connecting link. Okay, this jaws are modified into beaks, correct? Beak tail. Okay, tail is a bony and long, and forelimbs are modified into wings. Connecting between birds and mammals along the birds and the reptiles. Connecting in between birds and the reptiles. And the next one, which of the following is correct order of the evolutionary history of man? Correct order. The first pecking man, and the second one is the pecking man, not Homo sapiens. Pecking man, not Neanderthal. Pecking man, Caelid man, Neanderthal, Cro-Magnon, and Homo sapiens. Next is this Cro-Magnon, Homo sapiens should come. So it is the correct order of human evolution. Okay, next one. Look at this. Question number 17. Question number 17. The study related to the given below, the pictorial representation. So it is nothing but the evolution of human being. It is not sociology. It is not geography. It's not demography. It is anthropology. Human evolution. The study of human evolution is called anthropology. Which one of the following includes homologous organs? Homologous organs. The homologous organs, wing of butterfly, wing of bird, all these are analogous structure. Four limbs of frog, bird, and four limbs of rabbit. So it's useful for walking or locomotion or movement. This is the correct example for homologous organs. Next term, the chance of elimination of gene from a small population is an example of. In the question, if the term comes The concept which explain only about the small evolution, small population is genetic drift. They give large population, that is ordinary drift. Genetic drift proposed by genetic Sewell Wright. Okay, his name is Sewell Wright. Okay, proposed by Sewell Wright, bottleneck effect, and also founder's principle. And the next one, as per the theory of biogenesis. As per the theory of biogenesis, plants evolved from animals wrong. Animals have evolved from plants is also wrong. Spontaneous generation is also question number 20. That is also wrong. And present day organism have developed from pre-existing one is the correct answer. For this, question number 20, fourth answer is the correct one. 21, extremities tail ear are relatively shorter. The animals which are living in the cold area, they have shorter. The rule is called Allen's rule. It is called Allen's rule. The animals which are living in cold climatic conditions, they have a short tail. Short tail. So next one, Darwin's in his natural selection theory did not believe discontinuous variation. Right? He did not believe discontinuous variation. He believed parasite and predators natural limits, survival of the fittest, struggle for existence, overproduction, competition. All these concepts he believed, but he did not believe the discontinuous variation. Actually, variation is a continuous one. According to him, the variation occurs according to the environment. Next one, ancestor of Triceratops. Triceratops. That is the Triceratops. That is origin is a dinosaurs. Right? Have three horns inside the developed from Stegosaurus. Stegosaurus. Brachiosaurus is the largest one. Okay. Tyrannosaurus have very long neck length. Long neck. So next, we come to this big structure. Look at this picture. Darwin noticed the changing pattern features on which of the following island. This is not feeling well. This is easy one. This is Galapagos Island. He visited Galapagos Island. He took 5 years of expedition in his ship called HMS Beagle. Flippers of penguins and dolphins is example for convergent evolution. Convergent evolution, because the penguins are bird, dolphins are mammal. They are living in same environment, but the organ developed for locomotions from different organ, but the function is similar function. And the next question, the formation of life preceded by chemical evolution. The statement was given by this Oparin. Statement was given by Oparin and Haldane. Chemical evolution, the year 1928. But William Miller, they did experiment prove this concept in the year 1953. 1953. Next one, which are the following refer to the correct examples of organisms which evolved due to changes in the environment brought about by anthropogenic action. According to this, see this question, Darwin features of Galapagos Island which have evolved due to changes in the environment. This is a natural selection. But the question is anthropogenic action. They developed by human being. So herbicide resistant variety developed by human being. Drug resistant eukaryotes developed by human being. Man created the domesticated animals like dog. Right? This first one is a natural selection. Remaining three are anthropogenic. So correct answer is B. Correct answer B, not the first one. Next. So Question number 28. The ratio of methane, ammonia, hydrogen. 2:1:2. 2:1:2 is the ratio. And among the human ancestors, the brain size was more than 1,000. More than 1,000 cc is only in Neanderthal ancestors. Neanderthal, Cro-Magnon, and Homo sapiens. Remaining Homo erectus, Ramapithecus, Homo habilis. The brain capacity was less. That is less than 1,000. Less than 1,000 cc. The next okay, the Miller's experiment used methane, ammonia, hydrogen, water vapor, and closed temperature is 800 centigrade. In the closed flask, 800 centigrade. Tasmanian wolf resembles Placentral wolf. Resembles Placentral wolf. So this kind of evolution is called parallel evolution. Evolution is called parallel evolution. Both are having similar characteristics, but they are living in different area, different places. Next, the following use and disuse theory proposed by Lamarck. Survival of the fittest by Darwin. Apparatus Stanley Miller. Mutation theory Eupodemus. So 1, 2, 1B, okay, 1B, 2C, 3D, 4A is the third option. Sorry, second option is the correct one. And the next, this is 34. Question number 34. Which of the following experiments suggest the simplest living organisms could have originated spontaneously from non-living matter, from non-living things? So here meat was not spoiled, heated and kept. See, this experiment done by Francisco Reedy. And he found after boiling the meat is kept in the 3 different jars, and he could not find the development of any organisms from the bottles which were completely closed, diagraphically closed, and also closed with a string cloth. 35. There shows adaptive radiation resulting competition for food. Competition for food. Example, it is Darwin finches in Galapagos. So because of competition only they developed different types of beak, not Spicillania, DDT resistant mosquitoes. Okay, this also have the adaptive radiation, but not for food. Peppered moth, there is adaptive radiation because of the surrounding. Darwin finches represents not morphological variation. This is a geographical isolation. They developed the different peak modification developed in the different animals, including different habitats. Wings of pigeon, butterfly, and bat shows the evolution called convergent evolution, because these are analogous organs. These are analogous organs. These are analogous organs. So, which one of the following features closely related with the evolution of human? Not flat tail, not evolution, not flat tail. Flat tail is there in some. Okay. So flat tail. Shortening of jaws, not binocular vision. Okay. Closely related to evolution. Okay, there is a loss of tail. See, loss of tail is the correct one. And you see some more questions. You see that which one of the following phenomena supports Darwin's concept of natural selection? Development of transgenic animals? No. That is recently developed. Dolly was developed in the year 1996. Prevalence of pesticide resistance is the correct one. Okay, and the development of organs from stem cell. All these are actually, these 3 are the recent researches. So not relevant to Darwin. Occurrence of endemic species in South America and Africa is due to occurrence Occurrence of endemic species. They are very unique one in particular. That is due to not progressive evolution, due to continental drift. The movement of the continent that leads to origin of the endemic species in a specific area, including Australia. Primitive man was originated during. Primitive man originated during this Pleistocene period. Origin during Pleistocene period, not Miocene, not Holocene. So all these four periods of Cenozoic era. This is a Cenozoic era. Cenozoic era. Occurrence of endemic species in South America and Australia. Again, this is also due to continental separation. Continental separation. And random. So these are two opposing views about the origin of modern man. According to one, Homo erectus in Asia were the ancestors of the modern man. The study of variation of DNA out suggests African origin of modern man. Okay, which one is the correct one? See, remember this. The genetic variation were found more in African people than is Asian people. So the variation that is more in Africa than Asia. Greater variation in Africa than Asia. So that's why we consider the human being that is evolved from only the diversity originated in Africa, diversified to other continents. Next one, random genetic drift. Random genetic drift in the population probably results in random genetic drift that leads to increased more genetic variation. Not answer low mutation rate. They won't lead to large population size. That leads to high genetically variant individuals because of the genetic drift. That is concept proposed by Sewell Wright. Arrange the periods of Paleozoic era in ascending order. Paleozoic era cause the, you remember this, first Cambrian period, Ordovician, Silurian, Devonian, Carboniferous, and Permian. Right? All these are the different periods of Paleozoic era. Different periods of Paleozoic era. And 46. The process of evolution of different species in different geographical area, starting from a point, from a particular point. So radiating to other area, that is called adaptive radiation. Adaptive radiation. Example: Darwin finches, marsupial organisms. All these things you can write as an example. Which of the following best shows the common origin of man and the chimpanzees? Common origin of man. So the human being, of course, belongs to apes. Chimpanzee also belongs to apes. But the chromosomal number is also more or less very close to one another. But the chromosomal banding that only gives these two group of organisms are closely related to one another. Not the binocular vision, not clinical capacity, not the dental formula. And we come to this one. Be successful. Okay, evolution. Mutation must occur. Mutation occurs only in the germplasm that will be inherited. If mutation occurs in the somatoplasm, only the changes will occur in the particular individual, but not inherited to the next generations. Which one does not favor Lamarckian concept? Does not favor. Melanization of pepper knot is not a Darwinian concept. It is right. The lack of pigment here, that is the correct one, because they acquire the characteristics during the lifetime. Absence of limbs, that is also lost in their character. And the webbed toad is the advantage character that is developed in the aquatic organism. Melanization is actually natural selection, and also due to the environmental change it develops, that is not supporting the Lamarckian concept. And which one of the following variations temporarily they have nothing to do with the last or the next generation? This hereditary variation, of course, that leads to next generation. Discontinuous, and here the environmental variation. These are wrong. Okay, it is which are the following variations are temporary? Temporary is environmental variations. Okay, environmental variation temporary. Presence of gills that tadpole of frog indicates that fish evolved from frog-like ancestor. Wrong. Frogs will have gills in future. Wrong. Fish were amphibious in the past. Okay, wrong. Frogs evolved from gill ancestor like fishes. This is the correct one. So, dear students, I will send you this assignment questions to you. Go through all the questions, and suppose you have any doubt, you can ask me. Hello, students, are you hearing? Hello, students. Hello, if you have any doubt, you can ask me now. Do you have any doubt? Hello? Do you have any doubt? So you unmute your mobile. If you have any doubt, you ask me, and you have to prepare in your the next lesson also, human diseases. Okay, so all the best, prepare well, and you write the examination well tomorrow, and we have the NTA NEET examination on September 12th, right? So all the best. If you have any doubt, you can call me. Thank you very much. Sir, excuse me. Sir.